the Feindel Virtual Brain and Mind Seminar Series. My name is Boris Bernhard, and it's my pleasure to host the session today. Before we get started, please turn off your cameras and mute your microphones so that we don't interfere with the presentation. Thank you. So the lecture is co-organized by Bradislav Misic, Nathan Sprang, and myself, and we get help from Sasha Kelly from Neuro Events team. And our weekly lecture series, we try to feature early career investigators as well as established researchers that try to cover like the broad range of methods and interests that we do at the MNI. For today's lecture, it's my pleasure to introduce Dr. Denis Engemann, who's a principal investigator at INRIA and certified psychologist from Paris. And he's also a visiting researcher at the Max Planck Institute of Cognitive and Brain Sciences in Leipzig. Dr. Engemann obtained his PhD in psychology from the University of Cologne in 2015. As already mentioned, he's now a PI at INRIA, and he specializes in large-scale data analysis and predictive modeling to find clinically relevant biomarkers based on EEG and MEG techniques. Thank you very much, Denis, for giving the talk, and I'm really looking forward to it.
Thank you very much, Boris, for the kind introduction. It's a great pleasure to be here. Thank you. Good afternoon, everyone in Montreal. I will now share my screen. Okay. Okay, I think we should be good to go. I just have to arrange my windows here. Okay. Can you see my screen?
Yes, perfectly.
Can you also see the cursor of the mouse?
Yes, we can.
Okay, so if there is any hiccup or any interruption, Boris, please let me know. I'm not able to respond to visual signs at this point. I'm seeing my slides. Okay, so when looking into research in clinical neuroscience, we can end up in a situation that is depicted here. We have multiple brain signals, and we are trying to build models that relate these brain signals to clinical outcomes. However, when using high-dimensional data and multiple imaging modalities and other physiological modalities, we will end up with many models. And as a trend in the past decade, researchers have benefited a lot from and received lots of inspiration from machine learning approaches. And the hope is in this context that this practice can be simplified. So we have AI, we have machine learning, this is wonderful. So we can now pipe all those multimodal data into a predictive model and just combine everything into one single outcome, right? Isn't this wonderful? So the job is done. We can go to the beach and we can relax, and everything is settled, right? Well, not so fast. There are certain problems that will make us required to do a little bit more work, and gives us the impression that predictive modeling in clinical neuroscience is to some extent still a project that has to be realized. So first of all, very often the generative mechanism is actually not known. So some of you may be remembering this paper by Jonas and Cording from a few years ago, where the evil idea was to see if something can be learned about the inner workings of microprocessors by applying statistical methods, and the provocative answer was no. So what are we going to do with this? Well, maybe you will say it's not so important because we have machine learning, right? So we are just going to learn the causal structure somehow. I mean, what they were doing was just statistical modeling, and so it's going to be fine, right? Well, I have to tell you once more, not so fast, because unlike physics, where you can have a mechanical model, where once you know how to build your model, make a prediction, for example, the time of sunset tomorrow in Montreal, then you're good. So you can predict and you have the inference. However, in machine learning and statistical learning and any statistical modeling, we deal with black box models that are essentially surrogate models for a much more complicated underlying reality. If you want to learn more about this, with my collaborator and colleague Denis, who is also on Montreal at Mila and McGill these days as a professor, we have carried out, I think, a pretty large amount of simulations, around 100,000 simulations, in which we introduced model violations and noise and played with the ratio of a number of independent variables and really causally relevant variables in our simulation. And what we saw is that there was in many cases a strong divergence between the capacity of the model to predict new data and the capacity of the model to identify and isolate the right variables that were relevant in terms of classical null hypothesis significance testing. Well, but then you can say once more, okay, we don't care so much about the inference. We can use the prediction signal, right? We have cross-validation, and we're going to work with the prediction. It's going to be all right. But once more, not so fast. This time, Gaël Varocco is going to come, and he's going to tell you, look at the articles around you. In neuroscience, we are in a regime where we operate on relatively small data sets, and the methods that were borrowed from the machine learning literature are actually well working with larger data sets, much larger data sets. And what you can see in many cases is that as you increase the sample size, the predictive accuracy and the predictive success is also decreasing. Why is this? I think Gaël would say that this is indicating overfitting. Others say that this may be a problem of heterogeneity. These models may not be capable of finding a good surrogate for the real data generating mechanism. At this point, we don't know, but it is a real issue. And so what are we going to do with this? Are we doomed? Yeah, so the generative mechanism is unclear. It's not clear how to get inference. And then even the performance measurement can be corrupted. So what are we going to do with this? Well, so we are operating in high-dimensional data and in small data regimes. So what we need in order to move forward is to have good priors or to have a way to build effective shortcuts. Let me give you one example. Let's presume that we want to predict a precious clinical outcome. By precious, I mean that it's only rarely observed or that the data is quite expensive to acquire. Maybe we could think of some very advanced assessment of cognitive decline that takes many experts and complicated diagnosis, and the researchers cannot share the data on ethical grounds or on other grounds. And so what are we going to do? We cannot train a model on only a few hundred cases and hope that this is going to generalize. Well, what we can do is that we can search for surrogate measures. For example, we could build a model that is trained on predicting a widely available outcome, which then exploits the correlation with the outcome of interest. For example, age in the context of cognitive decline. And I think every one of you has heard about this because I think one of the most well-known surrogate biomarkers that has been established and that has been worked with in the past 10 years is the brain age, where the idea is that instead of looking at the passport age that counts the number of years since birth, we can also use a large normative database of brain images to compute a predicted age and look at this predicted age difference obtained from predicting the age and subtracting the passport age. And what you then get is an index that is, of course, relative to your population and asserts is a residual, where elevated brain age is indexing accelerated aging. So the theory goes. And in fact, there is now a wide body of research suggesting that a large brain age delta is not good for you because it indexes precocious mortality and has been linked with cognitive decline and other bad things that are unpleasant. However, interestingly, brain age is nowadays typically estimated using MRI, either MRI or fMRI or other imaging methods, but is less commonly estimated based on electrophysiology methods. And this is, of course, something that I find remarkable as someone who comes from an MEG and EEG background. Why is that so? Well, the hope is that using electroencephalography and magnetoencephalography, we can get a more direct measure of neuronal activity, because these methods allow us to actually observe neuronal events in a more direct fashion, not mediated through vascular effects. So is it a good idea? Shall we try to loop in the MEG and the EEG here? Well, do you think it's a good idea? I don't know. I mean, there has been now a long decade, even more maybe, in which there has been a strong historical emphasis on the similarities between MEG and fMRI. So for example, in the seminal work by Matthew Brooks and colleagues from 2011, the team has actually shown that it is possible to reconstruct large-scale fMRI networks based on source-localized MEG signals. Yeah, so that's great. So does that mean that this is all redundant? Similar work by Joachim and Markus Siegel from 2015 emphasizes on the frequency band specific correspondence of MEG and fMRI. So, okay, maybe this is redundant, or is it? Because perhaps there is more. If you try to emphasize on other things, you will find other things in this case, because the correspondence is anyways not perfect. And what we see in the last years is actually an emergence of research that emphasizes on the complementarity and independence of EEG and fMRI. Here, for example, in a study by colleagues from the MPI in Leipzig, Denis, who analyzed some of the Leipzig cohorts, Lemon and Live. This team emphasized actually on the point that EEG-based source localized signals in different frequency bands and fMRI-based connectivity don't have a strong overlap. So the source localization, there's lots of independent spatial information apparently. Similar paper from Max Nantwich and colleagues from New York on the Child and Mind Institute dataset emphasize the fact that the parcellation based on source connectivity from EEG in different frequency bands and to common parcellation scheme like the Euro Atlas give rise to strong to a lack of correspondence. So if you just compare the original EO Atlas with recomputed parcellation based on fMRI, you have a strong contingency, and it looks less impressive for EEG, in fact. So what do we do with this? So maybe we should consider acquiring some MEG and EEG data to see what is the added benefit of including electrophysiological signals in a brain age pipeline. So how do we do this? Well, one way would be to collect data, for example, at Neurospin, or you could also do it at MNI for sure. If there is a pandemic and you cannot collect data, one way to go about it is also to use Python, for example, because it allows you to just read in the data that have been recorded, of course. So this is a bit cheap. It's of course not replacing the problem. But in fact, there are nowadays curated data sets that allow us to benefit from recordings that have been done in the past. And if you do this, you get these typical signals. So in this case, this is from the MNE Python sample data set that comes with concomitantly recorded magnetometers and gradiometers and EEG. And this is how typical MEG signals look like. And then the question is, what kind of features shall we extract from the MEG? What you see here is data that I replotted from the Cambridge Center of Aging dataset, the CAMCAN, which I also analyzed in the studies that I will present today. And what you see here is the power spectrum of the MEG plotted on a double logarithmic scale. So here you see the frequency, here you see the log 10 of the MEG power. And I colored different averages by the age groups here, and you can see already that some interesting things are going on. For example, the alpha power seems to be lower in older people and higher in younger people, right? And that's great because I love alpha power. Yeah, I'm really interested in alpha power because in the past I have conducted work on disorders of consciousness, and to my surprise, I realized that the alpha band power, among hundreds of other features that you can push in a random forest, is the single feature that is most predictive of disorders of consciousness and enables cross-site generalization. So this is really robust and indexes something very fundamental about human consciousness and cognition. So we should definitely keep an eye on that. But then, of course, the EEG literature on aging is full of other things. So you have the alpha band peak, for example, as in the study by Babiloni. This is of course, not a comprehensive. Yeah. Then, okay, we have a high-tech device with many sensors. We don't have to just look at these averages. We can, of course, also compute topographies and look for spatial patterns that may reveal age-related changes in neuronal signatures. Then, of course, we have from the classical literature also these sort of power law dynamics approximated through 1/f fits, for example, classically in a linear model. Nowadays, of course, you can use FOOF and other tools. And of course, you have evoked latency changes and other things that have been reported in the literature. But I think a real important advancement that should be considered whenever possible is to consider the spectral energy, the spectral power in source space. Now you will say, yeah, okay, but is it really necessary? I mean, in my evoked response decoding, it doesn't really matter. And I will tell you, yes, this is not surprising, but it may matter when the phenomenon of interest is actually related to power spectra. So let's have a look at this. So how do we build an MEG-based regression model? Well, remember we have the signals that came out of the machine. So this is our X. And our objective is to predict a target from these MEG signals. This is our Y. And so what do? Well, we could use AI, right? So we don't care about anything and we just use AI. And you can potentially already predict that this is not what we are going to do here. So instead, we're going to think a bit harder and ask ourselves as neuroscientists, what do we know about our system? And indeed, there are a few things that we know already. So we know already that the MEG signal is generated by synchronously firing pyramidal cells in layer 4 and the cortex, a large part of it at least. So we can assume that there is a neurophysiological generator, which is of course unobserved because there are so many brain cells, we cannot see them. And importantly, we don't open the head of the people, we just see the extracranial signals. So what is happening on the way from the neural events to the extracranial signals? Well, we have field spread, which can be well approximated by the quasi-static formulation of the Maxwellian equations. And you can think that if this is a vector that contains all theoretically relevant neurons that can produce MEG signals, you have a big matrix here that is spreading around the fields and leading to the observations X. And many people think about this, but at the same time, it's important to think about the other branch here, which is telling you something about your outcome. So let's assume that we have a neural mechanism that produces behavior. I could move my fingers and we could assume that there is beta band synchrony in my motor cortex that actually causally produces movements in my fingers. So what do we do with this? First of all, we cannot observe the real thing, but what we can do is we can build a statistical model that allows us to estimate in a sense, latent sources. Let's assume that instead of the untractable Z, we have an S, which is a vector of sources. You can think of this like ICA components. It could be one brain region. It could be a set of brain regions that share a statistical fate in terms of variance. And you can put as many sources here as you can have sensors essentially on your extracranial measurement. And then what do you do with this? So here you get a type of ICA model where you say that the sources, the statistical sources, are spread with a mixing matrix, which does X, and then your job is to estimate both of them from the data. And many people look at this part. But I think the interesting details come actually from the second part here. We will also assume that we have a model that generates the biomedical outcome. And how does that look like? So this may look intimidating, but the idea is very simple. Since we have different sources here, we can assume that our Y is just generated as a weighted sum of these sources. In other words, this is just a linear model, a weighted average of the sources. And this is a general formulation, as you can see. So here is an F. What does this mean? For example, it could mean that the F is the log of the power. And this is exactly what happens when we are modeling age, for example, from the log power. So how does that cause problems? Let's say this is oops, let's say this is generating or Y. But what happens here is that you have a source which is transformed non-linearly, and then you have a linear operation all the way down here. And what happens is then, if you try to build a linear model based on the extracranial representations of the signal to predict Y, you are doing something bad, because the linear model cannot uncover what was going on here, because you are the nonlinear transform here. If this is not a nonlinear transform, for example, identity, it is fine. I mean, many users and researchers have observed that when performing evoked response decoding with linear models, it doesn't help much to go into source space, because in principle, the model can just uncover all these transforms that have been going on here. But my important point that I want to raise is that when you have a nonlinear component that generates your target, then you have to take care of it. Yeah, and one route to do so is source localization. Okay, so now we have an idea about how to build regression models with MEG and EEG, but how are we going to combine this now with anatomical MRI and functional MRI? So one way to go about this, especially in smaller data sets, is to use the stacking method, which was originally introduced by David Wolpert, and yeah, refers to the idea of fitting multiple models on the same data and combining those models, in a sense very similar to a neural network. And there has been a very interesting and creative usage of this kind of stacking approach in neuroscience and neuroimaging in particular in the past years. And here are a few studies that have used the stacking approach, for example, by Franz Liem and Teresa Carrer together with Daniel Obstock. And the idea is essentially that instead of filtering different models on the same data, you are building models that refer to different inputs. So say we have anatomical data, cortical thickness, we have functional MRI, and we have some MEG power spectra. Common choice is to have a first layer in which we are producing independent linear fits. And why we are doing this is because we have very high dimensional inputs, and we know from statistical decision theory that the expected out of sample error will of course grow linearly when we use a linear model and exponentially when we use a local model. So to tame this very high dimensionality, we have to do something about it, and a good way to go is to do a linear fit. This allows us then to forward for each modality the prediction, in this case the prediction of the age, and we can now introduce elements of local regression, for example by forwarding these age predictions into a random forest model, which can then learn a nonlinear function and combine in a very flexible way these modality-specific inputs and introduce elements of local regression on a much smaller dimensional data set. In this paper that we have published in eLife a couple of weeks ago, things looked a little bit more complicated in detail. I don't want to overwhelm you with this, but I want to bring to your attention that there is a table that describes the entire feature engineering, and I want to bring to your attention that the part related to the MRI essentially was a clone of the feature engineering done in this earlier stacking study by Franz Liem and colleagues. So this allowed us to address the central question whether predicting brain age from MRI and MEG can enhance predictive performance. And the way you have to decode this plot is that you look at the baseline, which is the MRI-based prediction. Then you see what's happening when you're adding MEG or fMRI, which is already not bad. So you're reducing the prediction error by roughly 1 year. And when combining both, you have a really significant advantage that brings you far that reduces your mean absolute error by more than 1 year on average. Okay, and you can perform rank statistic on those. These are cross-validation splits, and you would see that they are all parallel. There is a strong ranking effect here, that the better model is also in every fold, most of the time outperforming the model that on average predicts worse. Okay, great. We can predict age better, but does it mean anything? And I have some good news for you. We found indeed that although we just analyzed a healthy cohort here, we found some interesting effects. First of all, when combining multiple methods, effect sizes often grow. Here you see a Manhattan plot. This is not an effect size, but if you would look at the beta coefficient of a model that links the brain age to some cognitive external score, you would see that the effect sizes also often grow when combining multiple methods. And the other good news is that the loadings, if you look at the coefficients, suggest that brain age delta goes along with reduced fluid intelligence or more performance on the mini mental state exam, higher depression, and sleep complaints and other things. So very much in line with the interpretation of health relevant direction of variation. And what is important for the clinical translation is if you unbox the random forest model, the random forest layer, you can get some idea about what is going on. And we find actually that my favorite frequency bands, beta and alpha, are really important for the model, for the prediction of the model, as it, suggested by the variable importance. And this, of course, gives us some hope that we can carry that forward into the clinical application. In the interest of time, I'm going to skip this interesting result about missing value handling. I can just tell you that there is some good news. I did not talk about this much, but we can. One good thing about the tree-based models is that we can essentially unlock interesting options for missing value handling. For example, we can do a double coding here. We duplicate the input data, then we put on the missing values a large number and a small number. This will allow the random forest to condition on the missingness. And if there is information in it, old person is cannot go on the MRI any longer, for example. And then it can learn from it, and otherwise it allows us to not throw away data and combine everything. And we can show here that doing so, combining all the data with an opportunistic stacking model, is always worth it. And it doesn't give us any result that is worse than if we wouldn't have done it. This gives us the following interim summary. The MEG contains unique information on cognitive aging. I hope I could convince you about this. And at least you may have understood that I'm really a fan of source power. And third, tree-based approach in the stacking layer is interesting because it allows us to perform local regression, but also to get a flexible way of handling the missing values. Okay, wonderful. Hi-Fi device, 306 sensors, healthy population. Didn't you talk about clinical biomarkers, Denis? What is this? Look, when I'm in the hospital as a doctor, a friend can tell me, for example, things look a bit different. Yeah, do I need MEG and MRI to do source localization? Because we don't have MRIs in many patients who undergo anesthesia or who undergo EEG testing. So what do we do here? Can I also work with EEG, and do I need source localization? What do we do with all these things? And well, what if I told you that we don't really need the MRI? There may be a way to work around. Yeah. So how could that look like? One hint comes actually also from this movie. It's the Matrix. At this time, the covariance matrix, of course. Why is that? Let's look once more at these signals. Yeah, and you may realize that there are strong correlations in these signals, and some of them may be indexing functional connectivity or spatial extent of a brain source. Others of them may be due to artifacts like here, or due to the field spread. So the idea is that by representing the signal as covariance matrix, there may be some interesting procedures to unlock that can help us to reduce the effect of the field spread. So let's read in some MEG data, compute the covariance. You can do this actually, so here's the math. But usually I do it in Python, not by hand. We compute the covariance matrix, and now we can plot it. And what you see here, is this functional connectivity? Well, I think not, because what we see here is the broadband signal. So this is full of, you see there is lots of structured noise here. There are strong correlations, and these are not physiological patterns. So one thing that we can do already is we filter the data and we remove the low energy signals, low frequencies. We recompute the covariance, and now we see much more patterns here. Still, lots of things that you see here are due to the field spread and are not due to the neuronal events of interest. However, you can think actually of this, what we are doing here, filtering and then computing the covariance, as a way of computing power. Because in fact, if you just look at the diagonal here, this gives you a power estimate when you compute this on the bandpass filtered data. So I said I want to hack the matrix, and here I wrote some custom Python code. I think you can consider this hacking, to extract the diagonal, the diagonal, and make a topo plot from it. And you see that this looks actually quite physiological. You know, this could be a nice occipital alpha generator. And we will therefore use this representation as a baseline that we call diag, or diagonal in the forthcoming slides. Then let's go back to this time we will insist on this point that we use the covariance as a representation. And if you wonder about the indices here, the I is the xi is the subject I, okay? We are not representing trials here because we are not interested in single trial behavior. We are interested in cross-person regression. So the xi is the subject I. And this is work, conducted by my PhD student David Sabak and presented at Neuro's conference last year. And we also have an extended article in Neuroimage that is more targeting the neuroimaging business. Okay, so what do we do with this now? Well, one thing that we can do is that we compute a Riemannian embedding. So what is this? Let's have a look here. So here we have the CI, which is the covariance of a given subject. And here we have the C bar, which is the covariance averaged across all the subjects. And this little thing here is a whitening operator. And since it's a square matrix, we have to do the whitening from both sides. And then we can compute a matrix logarithm, which intuitively produces a relative score where the average of the covariance is taken out. And with this, also the average volume conduction. At the same time, this transformation is well known in geometry and has the nice side effect that the covariances which live in a curved space can now be represented according to distances in a Euclidean space. And this is very similar for the navigation problem that you have when you have to find a good way to fly from Montreal to Paris. So how do you do that? You don't look at the Mercator projection in 2D and draw the shortest distance. You take into account the curvature of the Earth. And this is also what we do here. And in our context, it has the nice property that it takes out the average volume conduction or the average field spread. So we compute this, and then we can just apply classical ridge regression, for example, on the transformed covariance matrix. And okay, this is wonderful. Can you show me that it actually works? And indeed, we have to deal with several model violations here. Some of them are less important than others, but as a proof of principle, we generated random matrices that obey to these generative models. And now what we can do is we can increase the distance from the identity matrix when performing the mixing operation here. So if A is identity, there is no mixing, all is perfect. If the distance from identity gets larger, we have strong mixing, and our diag estimator, which is just the naive power on the sensor space, it will get worse and worse and worse. And the Riemannian model is immune. That's crazy, no? No, it's not crazy because it's a property, a well-known property of the Riemannian geometry that it gives us this immunity to affine transformations, affine invariants. More importantly, we are dealing here with a situation where every subject has its own brain, and this leads to trouble because this means, in our theoretical terms, that everyone has his own mixing matrix. Yeah, and well, luckily, our participants are Homo sapiens, and from subject to subject, from participant to participant, the brain cannot be infinitely different. But still, there can be important geometrical differences, and hence you have individual mixing here. And when we simulate this process, eventually all the methods will fail. However, as we increase the noise on the individual mixing matrix, the Riemannian model dies later than spatial filters and naive baselines. Okay, wonderful. This is all in silico. This is all simulated. Can you really show me that this works on real data? So let's get back to the CamCam data and compute empirical benchmarks. And the short answer is, if we compute empirical benchmarks on the MEG sensor space using Riemannian embeddings, we really improve over the diagonal estimator. And we are getting pretty close to the source localization-based result. Okay. So when performing the same pipeline after computing the source time series, the performance doesn't get much better. In the eLife paper, we had additional features that improved the MEG beyond that, but essentially the Riemannian model gives us a performance that is close to the results that would have been obtained with source localization. Now, let's make an experiment. Let's apply minimum norm operator and run the entire benchmarks in source space. Will we get the same ranking? Keep in mind that the claim is that we can improve, that we can replace source localization with the Riemannian model. So if that's the case, and the answer is no. What you see now is that actually the diagonal model, which is just ignoring the cross terms, performs best. And that makes a lot of sense, I think, because the MEG operator did its job in taking care of the geometrical problem. Okay, so and, there are many more variables, and it's likely that by using a model that has more than the cross terms included, we just cannot really benefit from it in the setting. But the big picture view for me here is that essentially we can use the Riemannian embedding as a surrogate for minimum norm-based or source localization-based handling of the geometrical problem. Well, we still have the other translational problem. Now we have seen that it works for MEG, but will it also work for EEG? And in the study by David Sabak, we have also included the Temple University dataset with more than 1,000 subjects in healthy subjects, and performed the same analysis on 21 channel EEG. So 306 MEG channels versus 21 EEG channels. You think it will look different? Same? Well, it looks very similar. We have a very similar baseline in terms of predicting making random predictions, because the age range is very similar in the Temple Uni dataset. And importantly, we get really similar benchmarks here. So, of course, this can be a problem with n equals 1. We haven't looked at the other millions of data sets that are theoretically conceivable. But it gives me some hope that this is actually a way that we can pursue. When source localization is not possible, we can essentially use these approximations as shortcuts, and it can also work with EEG. There are some interesting trade-offs, of course, that we can think about, because EEG has sensors that are affixed to the head, so the effect of field spread may be more consistent. On the other hand, forward model computation in EEG is more involved, and the data may be more smeared. So there are some interesting trade-offs. If you want to get really an idea about the relative merits, we would need to collect EEG data and MEG data simultaneously and look in the same subject. What kind of sensor configuration and what kind of paradigm is supported here to say whether we can translate or not. So if you want to have a few take-home messages beyond what I have already told you during the interim summary, the point here is that we can actually obtain shortcuts that are statistically and mathematically motivated that can help avoid source localization. Okay. In the predictive setting, of course. We are not getting inference for that because it's very difficult to then interpret the outputs. This is active research. So we are really in the prediction paradigm. We cannot tell you what is the brain source, but we can predict from it. And the other good news is, if you're working in clinical neuroscience with this approach, we can actually also substitute the EEG for MEG, at least when predicting age. And of course, all what I'm saying here cannot be generalized beyond the kind of benchmarks that we performed here for age prediction, because it depends, of course, on your generative model. I think in the interest of time, we should come to an end. I just want to highlight that there are, of course, important resources. The MEG community is a friendly place. We are also friends with other toolboxes. Sylvanus may be chuckling now behind the video wall. When you come to Biomedical, you can see that MNE and Brainstorm and Field Trip are friends and are presenting together. But I'm speaking as a long-standing participant and contributor to the MNE community here today, and I'm of course highlighting the tools that I have contributed to and that I'm using. And this is the MNE software. But importantly, if you want to get inference out of your data and you don't want to do this kind of black box modeling, it may be interesting to clean your data carefully. And the good news is that machine learning can also be your friend for that. So we have different tools as satellite projects that can help you in automatically cleaning the data and automatically computing covariance matrices with the proper amount of regularization. And then, of course, you find satellite projects that can help you accessing large-scale data resources like the human connectivity or piping your data into R, for example, for more sophisticated or let's say custom analysis. And I want to conclude thanking all my collaborators and co-authors. I think there are more collaborators and co-authors to thank than you find on my slides, but I'm highlighting colleagues and researchers here who have contributed to the main results that I've presented today. Thank you.
Yeah, thank you very much, Denis. That was an excellent talk with really interesting theoretical as well as practical considerations on how to analyze MEG and EEG data. So for the question and answers, we now have some time. So you can either turn on your microphone and video and ask Denis the question directly, or you can use the chat box and I'll read your question to the speaker. So maybe to get started, I just have one question because so the eLife paper data looked fascinating and really nice. Congratulations on that. I just have a question from an MRI perspective. Which MRI features did you combine with the electrophysiological markers there for the brain age prediction?
Yeah, I should have spent more time with that table. I was a bit lazy in saying that we just cloned the pipeline of Liam and colleagues. But the essential things are the free surfer outputs. So cortical thickness, cortical surface area, subcortical volumes, and then functional connectivity computed on the fMRI time series with tangent space embedding.
Mm-hmm. For the alpha source power, would you what kind of physiological effect would you or underlying structural effect would you think contributes to these shifts in alpha source power with age that makes it such a good biomarker? Do you think this is also related to the atrophy or to connectivity changes?
I think this is an excellent question because the answer may not be so clear-cut. So forgive me if I may speculate. I think one part of the story could be related to what psychologists refer to as a compensatory mechanism. So it may be that in older people, you have a change in the structure of executive function. And of course, alpha is a very important brain rhythm that has been, of course, set famously by Busaki to produce a partial disengagement from environment, and that is important inhibitory functions. So this is one potential route to think. Another aspect is, of course, much more low level, and it's something that we have investigated in the Neuro image paper to some extent successfully with empirical simulations, where we try to understand how much information of the aging is actually present in the lead fields. And the answer is that half of the performance that you can reach using the Riemannian model can be explained by information that is related to the anatomy, because that's information that, independent of any activity, is already present in the lead fields. And I think clearly more research is needed to disentangle these factors.
Mm-hmm.
Boris, can I ask a question?
Absolutely.
Okay. Well, congratulations, Denis. Great job as always. Just maybe a little bit of a provocative question. Why did you bother predicting the age, which is just a number, right? Even brain age, if you take the biological, clock as a reference for, measuring this number, the actual, relevance, behavioral, cognitive relevance of age is actually very limited. Why didn't you try to predict basically something more related to behavior, like the, I don't know, memory scores or whatever was available in this CAN database?
I think this is a good question, and I think I could answer to this question on scientific but also personal levels and historical levels. And maybe we don't have the time for all these levels, but Yeah, I think one important point is that this is also a study in the construction of surrogate biomarkers, and the ultimate goal was to go beyond age and to look to look at populations at risk to whom we didn't have access at that time. I would have been very interested to look at neurodegenerative diseases, but I could not find collaborators. I'm also working with colleagues from the Lariboisière Hospital in Paris. I'm predicting postoperative with EEG, and we have limited data, and we are fighting with the data quality. So there was a strong interest in building a normative baseline model. And the convenient idea of the brain age is that you, similar to the historical ideas about mental age, about IQ, you build a residual model. You look at the population and you assume that there is some golden mean, some normal way of proceeding, some normal way of being, of aging, and then there is orthogonal to that some residual variance that can be linked to precautious or accelerated aging. And then the idea is, of course, if you have a number like age for almost any participant in any data set, you could unlock the great benefits of powerful function approximation machines that are enabled by machine learning. So you need lots of data for that, and that could be one avenue. And this is why we got interested in the brain age. But I agree that if you have a specific interest in a specific target, and you have a rich data set that allows you to approximate a good function, then you can of course go directly for it. But very often we don't have that. Very often we are in a setting where, say, we have 40 or 50 or 60 patients, and we want to say something about them. And one way to go would be to build a brain age estimator and then forward predictions and apply classical statistics on the output of the brain age estimator that could then allow us to discriminate different groups in a smaller clinical data set. I hope that
I guess what you call the brain age is not actually the biological age of the brain, but rather the actual functional state of this brain or capacity of this brain, regardless of the biological age.
Yeah, it's even more horrible than that, Silva, because you have also this problem that this is actually an exercise in population variance decomposition, if you want, because you're dealing with populations. So if you move from one population to another, the brain age delta that you look at in one person is always relative to the rest of the population, just like an IQ score that normalizes the test intelligence with regard to some norm that comes from a table by comparing the performance with the performance of many other people in the same age. It's very similar methodologically, and therefore also inherits the problems that come from psychology of intelligence.
And maybe one last more technical question. I think you demonstrate at the end very clearly using your elegant approach that there is very little added value to doing source modeling if you do apply this kind of deconvolution, smart deconvolution, so to speak, on the sensor level. So I guess my question is, but you also show that even with an imperfect source model, you do actually achieve similar performances. Is that correct?
Yes. So if you can do source localization because you have the person power, the people power to collect the data and to do the manual co-registrations, and go for it. This is wonderful. The idea is that if we cannot do that, there may be a way around. Spatial filters can work. I did not highlight them so much because we learned in the course of the study that Riemannian embedding is very robust. I did not show this today, but when you turn on and off different pre-processing steps up to a point where you don't do any pre-processing at all, spatial filters or other methods will break essentially. And I think that maybe one reason why the Riemannian method is so robust is that it has this matrix logarithm that of course applies lots of compression to the data, and you have these nice invariance properties. So practically what we see is that the Riemannian embedding is very robust to pre-processing options. And one way I think about it is also that it could be a very fast and cheap end-to-end pipeline without deep learning. Yeah? So you take your data, and you have a question and you want to see if there is some music in the data. So you just compute the covariances without any pre-processing. It takes a few minutes. You apply the Riemannian embedding and you build a predictive model, and you will get very close to what you would achieve by doing a very carefully engineered analysis. And on success, you can decide, okay, this is interesting, the prediction works. Now I can properly model it and can make that investment to go the extra steps and make the extra effort to get inference of the data. And do something about the predictors, which currently is not really easy with Riemannian embedding. It doesn't tell you really what is the importance if you apply linear models to the Riemannian embedding. At this point, it is not very straightforward to interpret that, although it may change in the future. This is active research in the community.
Okay, thank you so much, Denis. Thanks.
Thank you, Silva.
Thank you. I think we have another question from Danilo. Danilo, do you want to turn your microphone on?
Sure. If nobody else wants to ask a question, I just wanted to use the opportunity to thank for the interesting talk and to learn something about MEG. So I think the imaging neuroscience community has learned that no matter what you do with an MRI, age, sex, height, weight, BMI seem to be important sources of variation. Like, to what extent is that the same or not in EEG and MEG? I'm just curious.
I think this is a very good question, and I absolutely share your curiosity, Danilo. So I hope that I could tell you something about aging, and I think that aging is a very important modulator of MEG signals, either directly through changes in neuronal activity or through the anatomical changes, which are modulating the exposure of brain sources to the extracranial sensors. And I think that already arguing based on the lead fields, there is, of course, an important nesting. Statistically speaking, male persons tend to be larger on average than females, and this can also lead to different sizes in the head. So, of course, there's lots of overlap, but there's a very low-level possibility that these changes in head size will also affect your measurements in the geometry. Then about the functional changes, I'm not very sure about that. I think that the gross effects that you will get with MEG or EEG are always related to the geometry and the exposure of the brain regions in geometrical terms to the sensors. But I think these are interesting questions that would deserve further investigation.
Thanks.
Hi, Christoph.
Hello.
Hello, Christoph.
Very nice talk. I really enjoyed it. I will have one question regarding the data that you used. I'm not so sure if you can just clarify if those data, because you focused a lot on alpha, if it was eye closed or eyes open, or if you looked also at the level of drowsiness of the subject or things like that, because all this will largely affect the amount of alpha.
Yes, for sure. And in the CAN we don't have any estimate of the drowsiness or of the vigilance. And there are, of course, emerging data sets that are smaller, but specifically focus on vigilance and arousal. And in my work on disorders of consciousness, of course, the arousal component was really what we picked up with alpha as an index of residual consciousness. But in the CAN we really don't know, and I agree with you that this is an important confound factor, and it may add another mediator to what we observe. It may be that you have more drowsiness in people of a certain age range.
Okay. Was it eyes open or eyes closed within those database?
Eyes open, as far as I understand.
Okay, thank you.
Yeah, thank you all for the questions, and thank you, Denis, for this fantastic talk. It was really interesting and exciting. So I guess you'll be available for follow-up questions from the audience if ever there's somebody who has another question, so they can just shoot you an email and get in touch.
Absolutely.
Okay, very cool. So thank you all for joining, and see you next week.
Thank you, Boris. Thanks everyone, and I hope next time we can yeah, next time I give a talk, I can actually come personally to Montreal.
Yeah, come in the summer.
Yes. Okay.
Bye-bye.
Have a nice afternoon.
Bye-bye. Cheers.